Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
In the year 1951, Rosalind Franklin's research culminated in the paper titled Molecular Configuration in Sodium Thymonucleate. This work addressed a central challenge within the burgeoning field of molecular biology to ascertain the elusive, fundamental structure of deoxyribonucleic acid, or DNA, the very essence of heredity, the architecture of life itself, hinged upon deciphering this complex molecular arrangement. The paper's primary focus centered on the arrangement of the phosphate backbone within the DNA molecule. Franklin sought to understand how these crucial components organized themselves within the overall structure. At the heart of Franklin's contribution lay the presentation and initial interpretation of X-ray diffraction patterns obtained from crystalline fibers of sodium thymonucleate. These patterns, data embodied in the now iconic photograph 51, unequivocally suggested a helical structure. The paper detailed the precise physical parameters of this helix, revealing crucial information regarding its pitch, diameter, and the number of nucleotide residues per turn. Furthermore, the paper recognized the importance of water content, suggesting that varying degrees of hydration influenced distinct structural forms of DNA, later classified as the O and B forms. The O form appeared under drier conditions, while the B form emerged when the DNA was more hydrated. The interpretation of these complex diffraction patterns posed significant intellectual and technical hurdles at the time. The data presented a profound enigma, demanding a sophisticated understanding of crystallography, chemistry, and physics to unravel. One of the most significant challenges involved determining the precise location of the phosphate backbone. Was it positioned on the inside of the helix, cradled within the structure, or on the outside, readily accessible to the surrounding environment? The answer, crucial for understanding DNA's biochemical behavior, remained elusive. This work was situated within a highly competitive scientific landscape. Several research groups globally were vying for the honor of solving the DNA structure. This environment fueled innovation, but also introduced tensions and complexities in the sharing of information. It was within this intense atmosphere that Franklin's research took shape, marking it as a key player in the race to understand the molecule of life. Franklin's work did not arise in a vacuum. Her approach was informed by earlier investigations. Prior studies employing X-ray diffraction on fibrous materials by researchers like J.D. Bernal and William Asbury illuminated the potential of the technique. Their work paved the way for Franklin's methodological rigor. Furthermore, the theoretical framework of crystallography, with its principles of diffraction and spatial analysis, developed by Sir William Lawrence Bragg, provided the essential mathematical and physical tools necessary to interpret the complex patterns. The immediate impact of Franklin's work cannot be overstated. Although the paper did not make explicit mention of the fact at the time, the data within it became a critical catalyst for the creation of the famous Watson and Crick model. Franklin's meticulous data, particularly the X-ray diffraction patterns, provided Watson and Crick with crucial clues. They were provided access to this data through Maurice Wilkins, a colleague of Franklin's. The structural parameters derived from these patterns directly informed their model-building efforts, ultimately leading them to propose their now globally recognized double helix structure. The central claim of the molecular configuration in sodium thymonucleate paper was that X-ray diffraction data supported a helical structure for DNA. This claim was subsequently substantiated by the Watson and Crick model. The discovery of the DNA double helix, a structure of two intertwined helical strands, was a landmark achievement. The work also correctly identified and characterized the O and B forms of DNA. The structural variations that depend on the level of hydration, representing a further key piece of the puzzle. The X-ray diffraction patterns, central to the paper, are still considered a cornerstone in the understanding of DNA's structure. These patterns hold the key to unlocking the mysteries of the molecule. They provide a visual representation of the molecular arrangement. The importance of understanding DNA's structure is fundamental to modern biology, genetics, and medicine. The structure informs everything from the study of gene expression to the development of new treatments for genetic diseases. The helix unveiled by Franklin's research, alongside the work of Watson and Crick, powerful testament to scientific Rosalind Franklin's 1951 publication, Molecular Configuration in Sodium Thymonucleate, provides a pivotal insight into the structure of DNA, revealing its helical nature and laying the foundation for understanding its various configurations. A careful analysis of the experimental methodology, specific findings, and lasting impact of this work illuminates its significance. Within the history of molecular biology, the paper's interpretation of X-ray diffraction patterns obtained from DNA fibers represents a cornerstone of its contribution. Franklin meticulously analyzed these patterns, which arose from the scattering of X-rays as they passed through the ordered structure of DNA. By measuring the angles and intensities of the diffracted rays, it became possible to deduce information about the molecule's repeating units and overall shape. The presence of distinct layer lines in the diffraction patterns provided initial evidence of a helical structure, the vertical spacing between these lines being inversely proportional to the pitch of the helix. Measurements detailed in the paper allowed for estimations regarding their helical parameters, specifically the pitch, diameter, and the number of residues per turn. These measurements, derived from precise analysis of the diffraction patterns, suggested a repeating structure with a pitch of approximately 34 angstroms and a diameter around 20 angstroms. The paper proposed that 10 nucleotide residues lay within each turn of the helix, a critical detail that aligned with subsequent modeling error. A crucial finding was the observation of 2 distinct forms of DNA, labeled A and B. Crucially, the paper indicated that the transition between the A and B forms was hydration dependent. The A form, observed at lower hydration levels, exhibited a more tilted arrangement of the bases relative to the helix axis. Meanwhile, the B form, prevalent under higher humidity, displayed a more perpendicular orientation. These observations highlighted the conformational plasticity of DNA and the importance of water molecules in shaping its structure. Despite the groundbreaking progress, the paper also revealed the challenges Franklin faced when determining the precise location of the phosphate groups. The diffraction data alone could not definitively establish whether the phosphate groups resided inside or outside the helix. Speculation suggested the phosphate groups were on the exterior given their hydrophilic properties, a hypothesis later substantiated by the double helical model. The experimental methodology described in the paper involved meticulous preparation of highly ordered DNA fibers. Long DNA strands were extracted and carefully aligned to create a semi-crystalline sample. This alignment maximized the coherence of the X-ray scattering, producing sharp diffraction patterns, essential for accurate analysis. The setup involved directing a beam of X-rays through the DNA fiber and capturing the resulting diffraction pattern on photographic film. Data collection involved careful measurement of spot positions and intensities, which were then used to calculate interatomic distances within the DNA molecule. Franklin's work had an indirect, yet profound impact on Watson and Crick's model building. Maurice Wilkins, a colleague at King's College, shared Franklin's data, specifically photo 51, with Watson, the data serving as a crucial piece of evidence that informed their construction of the double helix model. This shared data definitively confirmed the helical structure and provided critical dimensions of the molecule. Though Franklin did collaborate directly with Watson and Crick, her experimental findings were foundational to their theoretical break. The subsequent confirmation of the DNA structure revolutionized fields such as genetics, molecular biology, medicine, and biotechnology. Understanding the structure of DNA provided a framework for comprehending DNA replication and protein synthesis. This understanding has also enabled advances in genetic engineering, gene therapy, and personalized medicine, significantly impacting human health. The lasting significance remains evident, the paper standing as a crucial evidentiary underpinning in deciphering DNA's structure. The ethical considerations surrounding the use of Franklin's data, particularly the matter of recognition for her work, remains an area of ongoing discussion. This consideration extends to the broad dynamics surrounding women in science. The paper and its legacy resonate throughout structural biology, continually influencing future understanding of the molecular basis of life. It represents a critical milestone in the progression.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.